Well, good day, everybody, and welcome to the Neuroscience edition of 5-Minute Psych. In this 5-minute segment, we'll look at neural communication, and in doing so, we'll first look at how a signal is sent within a neuron with the action potential, and then we'll look at how signals are sent between neurons with neurotransmitters. I hope you recall the resting potential of a neuron and how positively charged sodium ions, which are concentrated outside of the neuron, are being drawn inside the neuron by both electrostatic pressure and the concentration gradient. But sodium can't get in the voltage-gated sodium doors. They're locked, at least until a certain voltage is reached within the neuron. So what do you think would happen if those sodium doors suddenly opened? Well, this is what happens with the action potential. Suddenly, the sodium doors unlock and sodium rushes inside the neuron, swinging the charge away from minus 70 millivolts toward the positive. The charge in the neuron gets as high as plus 40 before the sodium doors lock again and the cell works to expel the excess sodium. Because the inside is now positive, potassium is also kicked out. But because potassium travels so freely through the doors, the neuron ends up expelling too much potassium. As a result, right after the neuron fires, it ends up more negatively charged than it was at rest. This means that a neuron needs a rest right after it fires before it can fire again. This design ensures that once those sodium doors open, what happens next will always be the same, and so each action potential is identical to the previous one, with the same intensity. This is called the all or none principle, meaning a neuron either reaches threshold and fires or not. It also ensures that an action potential could never travel backwards, since the area of the axon behind it is more negative than before. Some of you may be wondering what makes the sodium doors suddenly open in the first place. Well, you have to remember that the dendrites of each neuron could be receiving thousands of signals from other neurons at any given moment, and the cell body's charge changes as it processes all of those inputs. Some of those inputs swing the neuron in the positive direction. Those are called excitatory postsynaptic potentials, or EPSPs. Others swing it in the negative direction. Those are called inhibitory postsynaptic potentials, or IPSPs. If at any moment the neuron's charge changes from minus 70 to minus 60-ish millivolts, that is like the key to the sodium doors. Threshold is reached and the doors open. This would happen if the neuron received more EPSPs than IPSPs, enough to push it to minus 60 millivolts. So far we know how a signal travels within a neuron when it fires an action potential. But how is this signal transmitted to other neurons? Well, if we zoom in to see what happens when the action potential reaches the end of the axon, called the axon terminal or terminal buttons, we see that it causes these small balloon-like vesicles, called synaptic vesicles, to open. These vesicles are filled with neurotransmitters, or chemicals that can affect the next neuron. When the vesicles open, the neurotransmitters float in the tiny gap between neurons, called the synapse, and some of them land in receptors in the next neuron. Once there, these neurotransmitters affect the ion-gated channels on the next neuron's membrane, and so on. There are hundreds of different neurotransmitters, but some key players include glutamate, the most widely distributed excitatory neurotransmitter that's used by most neurons in the nervous system. GABA is the most common inhibitory neurotransmitter. Acetylcholine is involved in memory and muscle activity, and dopamine is used with positive emotions and voluntary movements. Serotonin influences mood, eating, and sleeping, while norepinephrine is important for mood stability and arousal. Now, a neurotransmitter would impact the next neuron as long as it stayed in the receptor, and many drug treatments work by increasing or blocking these neurotransmitters. Normally, our system uses reuptake to remove the neurotransmitters from the synapse. Yes, our brain recycles neurotransmitters. We just looked at how and why a signal is sent within a neuron with the action potential, and how this causes neurotransmitters to be released that allows this signal to travel between neurons. Now, aren't you glad you had 5 minutes?